Okay, good morning, everyone.
Good morning, Asha.
Mm.
Good morning, Asha.
Yesterday, I required my students and arranged the homework, reviewed the structure of the nucleic acid. Yesterday, we studied the chapter 1, the gene. This definition must understand. Maybe the molecular biology beginning from here. So, what is the gene? Gene, a stretch of the continuous DNA sequence. This is the sequence coding protein or RNA. It's very easy. Continuous DNA sequence. This sequence coding, encoding the certain protein or RNA. Okay. Today we study the other contents about the gene. The first here, you can see here, at least there are several kind of the major definition. Major definition. The first you can see the splicing gene. Splicing. What mean? This coding DNA sequence, it is uncontinuous. Here you can see this. My students must remember the before study this here. Study another definition, structural gene. In the DNA sequence in the gene used to transport cryptid produces the specific RNA molecule. This DNA sequence, it is the structural gene. You can see according to the base pair, and produces the RNA. A paired the U, T, T paired the A, T paired C, C paired G, and according this kind of the principle and produce the RNA molecule. This sequence we defined the structural gene. This, do you understand now? My students, this, do you understand now?
Understand, Asha.
Okay, in the genes, you can see the DNA sequence derived to synthesize RNA sequence. And this process you pay attention to. According to the base pairs principle, produces the RNA molecule. This sequence, the totally sequence in the gene, coding according the RNA sequence, this DNA sequence, it is the structural gene. Maybe some students maybe think, except the coding sequence in the gene, including other kinds of the sequence, yes or no? Yes. So later in this chapter, we studied all kinds of the regulatory sequence. Regulatory sequence, you can see this sequence level transcripted. Only regulated the relative sequence transcription. In fact, it's very easy. It's very easy. this thing is the DNA in the genes, the DNA sequence coding the RNA sequence. This sequence we give names structural gene. Okay, so in the eukaryotic organisms, all the times contain the spatial gene. This coding sequence, the structural genes sequence, all the times it is a discontinuity. Here you can see, like the PPT, you can see here, it is the model about the you can see here the blank sequence, the white, blank, white, A, B, C, D, E. Hey, you can see this letter, you can see up, you can see A, the small a, the B, small b, C. You can see here, all in B and A, the letter A, B, C in the future, encoding the protein. The smaller letters A sequence, smaller letters B sequence. These two sequences don't take part in transcripted and encoding the paper chain. So these things you can see coding the paper chains sequence, it is discontinuous. Discontinuous. So at that time we see this kind of the genes give the name splicing gene. In this PPT, you can see the down of this PPT, you can see here listed the agar bovines protein. You can see the 1, 2, 3, 4, 7, 8. 8 sequence marked with the black. You can see this 8 DNA sequence and last all in take part in coding the agar bovines protein. The A, B, C, D, E, F, G. This 7 sequence. All in transcripted produced the RNA. At that times, immediately splicing and deletion and destroyed. All in 1, 2, 3, 4, 5 stage, 7, 8. This 8 sequence coding the paper chain. So you can see how I list the shot and the robots. These two famous scientists find this kind of the gene and acquires 1993 Nobel Prize. And find in the eukaryotic organisms, you can see the structure genes all the times discontinuous. Here, my students write it down and remember In the eukaryotic organisms, the histone protein, the structural genes, it is the continuous. This pay attention to. Maybe my students studied, you can say, the structural genes of the eukaryotic organisms. All the times is discontinuous. This is wrong. Except and to not find. And another things, maybe students think in the prokaryotic organism. How about the structure? In the prokaryotic organisms, the structure genes, it is the continuous. Never produced the splicing gene. And here, exception must also remember, in the eukaryotic organisms, and to not find the histone protein, the coding sequence, it is continuous. Okay, two definition. The first, structural gene, and the second, splicing gene. Maybe here, down of this PPT, you can see here exon, intron. What's it mean? In one genes, you can see the coding peptide sequence. According this sequence, at the last left code left and encoding the peptide chain. This kind of the sequence, we give the names exon. And A, B, C, D, E, F, G. This same sequence, you can see in the future, you can see deletion, splicing. This kind of the sequence, we give names intron. Okay, and Legator definition, overlapping gene. What it means? You can say overlapping genes are defined as a pair of adjacent genes whose coding region are partially overlapping. Let me say here. You can say here. You can say these models. The figure B, you can see the gene A, B, C, D, E, F, G, H, G. All of names you can see. For example, you can say the B. The gene B is contained with the gene A. The gene B sequence overlapping in gene A. And another kind of the overlapping like this. I Let me take a photo and send it to all of you. Okay, my students see the Jingding talk. I send to you one figure. You can see here. You can see. Do you see clear?
Yes, 老师。
These two lines means one DNA molecule. You can see the double helix structure. You can see the gene A. You can see from this point begin to transcripted. The structure genes you can see transcripted and according the RNA synthesis and produced to the TER termination point. And here you can see the B genes, you can see contains the A gene, partially overlapping. And another thing, you can see the down lines, you can see the gene C transported another direction. And you can see gene A and gene C partially overlapping. Okay, so this you can see this kind of the phenomenon may be presented in the virus and prokaryotic organisms is normally. Certainly, in the eukaryotic organisms also contain this kind of the phenomenon, but the frequency is little. This is the overlapping gene. It's very easy. Overlapping partially sequence is okay. And pseudogene. Pseudogene. What is pseudogene? Maybe you study the psychology or physiology. Maybe you studied the globin. Last semester, you studied the biochemistry. You studied the structure of the protein. We also study, you can see, for the person during the development and the growth, you can notice the hemoglobin composition may be changed. Why? Because during the growth, different stage, different globin protein produced. So at that times, at the beginning, for example, the two peptide chain transcribed and produced, and choose the adult, you can see all in alpha and the beta. Why like this? Because later the structure genes lose the activity and no longer expressed in the cell. Why like this? And to not find maybe these genes may be modified, methylations, and so on, and make this sequence lose the function and never expressed again. This kind of the structure genes gave the names of the pseudogene. This maybe you combine the globin protein equation easily understand. And another one, jumping gene. This is a very interesting topic. At the beginning, the scientists presented this topic, and maybe other scientists think this is wrong. Maybe one DNA sequence, one DNA molecular you can say destroyed, just produced this kind of the sequence. What it means you can see in the genes, some sequence can be moved from here to there in the DNA molecular, in the chromosome. You can see a segment of the DNA that can becomes integrated at many different sites along the chromosome, especially a segment of the bacterial DNA that can be translocated as the wool. Why like this? I study this person, this lady, make clean talk, Barbara. This doing scientific research focused on the coin. You can see here this figure. The coins you can see some coins particle you can is the red, some is the white, some is the partially red, partially white. Why like this? This lady doing some famous research and find why like this. Doing some research and find you can see and find, for example, the DNA sequence. You can see maybe in the chromosome 9, you can see here. Look at the upstreams of the SH, BZ, WX before. This coins is a red coins particle. When this partial sequence may be integrated to the here, it is insert to the SH location. At that times, you can see this coins without color, white. So at that times, these ladies at that times presented that in the journals having some spatial sequence can be moved the location and integrated to different location. And that times many scientists think this lady may be crazy. All the times we lose the DNA molecular you can see have good property and keeps the stable. Like this, some sequence you can see can be moved from here to there, from there to here. Like this, stability come from where? And later, especially doing some scientific research focused on the bacteria and find this kind of the phenomenon. In the genome, have some spatial sequence transposing can be moved from here to there and integrated as a different location in the chromosome. This is another kind of the genes, jumping genes. Here, the last definition I listed in this the chapter 1, the housekeeping gene. Maybe this is the easy understander. How to understand it? Because in the organisms, all kinds of the cells you can see expressed there and keep the continuous expression gene. This is the required for all kinds of the cell for the maintenance of the basic cellular function. For example, histone. You can see in all kinds of the cell you can see required the histone and produced the is bonded with the DNA and produced the chromosome. In the eukaryotic organism, you can see the histone. It is the requirement for all kinds of the cell. So this kind of give the name housekeeping gene. If the cell deletions the housekeeping gene, every kind of the housekeeping gene, this cell must die. It's very easy to understand. For other kind of the cell, for example, the cell want to living needed the ATP. Last semester we studied, you can see in the cell produced ATP, mainly come from the oxidative phosphorylation. And you can see here refer to stable kind of the design. This you can see all of the teams like this give names housekeeping team. Coding reversely other teams, for example, different cells you can see express some kind of the gene. For example, red blood cell you can see the hemoglobin. But for the muscle cells, other cells you can see many kinds of the cells you can see without the hemoglobin genes expression. This kind of genes we give names luxury gene. You can see luxury genes coding for special cells products and is expressed abundantly. These are tissue specific or organ specific, which means they are not expressed in the cell. They are not constantly expressed only when their function is needed. Example of luxury genes is the plasmid of the bacteria and the genes coding the heat shocking protein. For example, hemoglobins in the red blood cell like this. All of these kind of genes give names luxury gene. Okay, the first class we studied the last contains what is the function of the gene. It's very easy. The genes storing genetic information. Maybe students think this the function similar to the DNA. Yes, because the genes sequence it is DNA sequence. So also storing genetic information. Genetic information based on the different base sequence. And the second, transmission of the genetic information. You can see all the genetic information is transmitted to the protein cells stably and forcefully through the replication. And last, act as the template and replicated produce the offspring steals gene. Template for the gene's expression. So this three factors, almost similar to the DNA function. Okay, we rest 10 minutes.
OK，老师。
嗯。
OK，老师。
Okay, come back and quickly finish this part. The first, for the genes, I use the model figure. Like this, you can see. All the times for one genes, you can see, including the regulatory genes and the structure gene. Structure genes coding the RNA or peptide chain, and regulatory genes regulated the expression of the gene. If the genes without the regulator gene, for example, for the without the promoter sequence, these genes never expression. So simply summarize, one genes equal to the regulatory sequence added structure gene. For the further protocol design or various have another kind of the things under the same one regulated sequence. Maybe regulated stable regimes same times expression. Like this, you can say the Jacob and Molod in the 1961 and presented the overall zero and acquires the Nobel Prize. You can see, and another thing is I just explained that you can see. I tell all of you, you can see in the structure genes, you can see the region of the DNA that coding contains the coding sequence which make up the matrix data among. The according DNA sequence, it is the exon. Intron, you can see here, it is the intervening sequence between the coding sequence. They will also be transcribed, but will be removed to produce the matrix among. This, it is splitting genes. You can see the coding sequence, it is the exon. The low coding sequence, it is the intron. This is easy to understand. Like this, you can see from the promoter, it is a regulatory sequence. You can see from here, the transported originated point began to transport the message RNA. Precursors RNA, you can see here, the exon left, the intron deletion, exon 2 left, and the intron deletion, and the last produced the matrix among. The matrix among, you can see the coding the protein. Another thing is you can say regulations sequence. In the genes, the according regulatory sequence takes the action. We give the names. It is the cis acting function. And another Another thing, you can see here cis actin elements, the DNA sequence that normally affect the expression of the gene, physically forms the same DNA molecule. Usually, low protein is encoded. For example, the enhancing silencing sequence is on most in the side or introns of the gene, such as the promoters, terminators, enhancings, the multipulated genes, it is on. And another thing, according to this, it is the trans-actin factor. All the times, it is the protein. And coding there from another side of the site, the gene, coding the thing, the protein. The protein may be aggregated and bonded, may be action to the certain gene and regulated the gene's expression. This kind of the protein gives names trans-actin factor. You can say bonding it is target loaded the same DNA molecule, such as the transporter factor. This way, in the future, we studied the transcription. You see, the all kinds of the transcription factor affect the transcription. You can see here. This is the for the regulatory. Here, I don't regulate translation. You can see here 调控序列. It is the regulatory sequence. For the downstream gene expression, the action, it is cis actin elements. You can see another side of the genes, you can see produce the protein. Protein A, you can see act to the gene B's promoter. The gene B's regulatory sequence and produces the function. It is the trans-actin factor. Certainly, you can say protein B also can be trans-act to the protein A, or gene A. Is easy understand. Another side, maybe you read some more textbook, maybe you read some more papers, maybe you use all the times two direction my students pay attention to. The upstreams, downstreams, the starting point, originated point. Here, all of this pay attention to. Here, start to transport the RNA. This place here gives names starter point or originated point. Originated point. Here we marked with number. It is positive 1. And the second base, it is the positive 2. And 1 by 1 to the downstream. We use the positive number marked the location. And the reverse direction. From this point, the positive 1 and the upstream, the first, it is the negative 1. The upstream's sequence, we marked the positive number. Positive, the negative 1, negative 2, negative 10. So these two directions, we use the upstream downstreams use the positive number marked the location and the negative numbers marked the location. Here, my students remember and understand the zero label used to mark the site in the teams. Okay. Maybe students, how to do the positive one, the originated point. My students remember, and several years ago, I meet one student think, from the promoter. Here, the first nucleic acid, nucleotide, the base, it is the positive one. No, my students remember, we began to transcribe it, produce the RNA, where it is the positive one. Okay, and another words you can see regulatory sequence of the proteolytic organisms. So here for the eukaryotic organisms and the prokaryotic organisms, you can see different regulatory sequence. You can see in the prokaryotic organisms, you can see here the promoter. And oriented to transcription. Here, give the signal, give the fitable condition, and the transportees can be bounded and begin to transcription. And another one, terminator sequence. Here, provided the termination signal for the RNA synthesis. And third, you can say identification and bonded the repressor protein in manipulated elements. This during the regulation of the expression of the genes, we studied this. You can see in one genes, some sequence you can see can be recognized by the repressor protein. Let me see. Oh, here you can see for the lactose opera. You can see here marked with the purple colors sequence. You can see in the future can be bounded with the CAP protein. The P it is promoter. Here all you can see manipulated sequence can be bounded with the protein. So here give names. You can see it is the representative can be bounded and identification and bonding. Here you can see regulatory sequence. Here in the promoter, you can see have the several kind of the sequence. For example, here you can see the negative 10 region. And to know in the prokaryotic organisms, you can see find this kind of sequence located in the about negative 10 point region. So give the names negative 10 region. You can see the composition. You can say TATA. So all the times this certain sequence also gives another names TATA box. And this sequence first find by the probuller, this scientist. So this sequence gives another name, probuller's books. Here my students are all in the Periodicalism can be give this last name, Probulus books. Why? Because the Probulus never doing scientific research in the eukaryotic. So later we begin to study this kind of things also find it in the eukaryotic. And another thing you can see, located in the negative 35 region, you can see have the DACA such conservative sequence. We give names negative 35 region. Give the names DACA books. You can see all kind of such sequence together and forms this region. And the function can be promoted the originated the transcription and started the transcription. So give names promoter. And here you can see just with this Jacob and Bollard in 1961 presented the lactose opera. Like this, the composition. Because they were it is the three kind of the enzymes. Take part in the lactose metabolism. You can see the promoter here contains the Probulus books, contains the negative 35 region, and to not find manipulation sequence and the CP bonding site.喂，好，我马上下去。Like this, you can see here represent protein, the coding genes, represent gene. Produce the mRNA, produce the represent protein, and you can see low lactose, and that times you can see represent protein. You can see never bonded with the manipulated sequence. The RNA bonded to the bonded with the manipulated sequence, and RNA polymerase never transcribed the downstream the structure gene. At that times you can see when with the lactose, and sometimes you can see, and that times you can see can be induced the represent protein change the structure. At that times can't bonded with the manipulated sequence. At that times downstream structure gene transcribed. Another thing you can see here, low glucose. CMG is very high, and that times you can see CP protein enhanced and bonded to here, bonded to the CP protein bonding sequence, and enhance the transcription. So here, this is PPD. Another PPD you can see listed here and summarize the regulatory sequence in the eukaryotic organism. Here, must pay attention to promoter, master, upstreams the positive one. Located is the upstream, and enhancing, and to now find maybe in the structure genes, maybe upstreams, maybe is far away of the upstreams of the gene structure gene. Enhanced genes, all kinds of the sequence have their function, enhance the expression, enhance in the transcription of the gene. This kind of sequence give names for these genes. It is enhancing sequence. And another, it is silencer separation, the silencer sequence. You can say silencer separation the genes expression. And another thing you can say, maybe in the future, maybe in fact, all of you understand that you can say one is the genes family, another one genes cluster. Means you can say one genes family, it is a state of the stable family gene formed by the duplication of the single originated gene and general with the stimuli by chemical function. And another thing you can say, this you pay attention to the biochemical function is similar. The composition is similar. But the genes cluster, physically you can say labeled each other. You can say a gene cluster, it is the group of the closely related genes that all coding the same function. Variation on the same function. Yeah. Together and finish this function. Together. Yeah. And the genes family, you can say one can be finished this function. Singly can be finished. But here genes cluster, you can say all the times may be labeled each other in the genome. Another thing is you can say all of them together and finish the one function. Meaning here is the physical labeled each other. Here you can see, for example, globin. You can see the alpha, beta, gamma, the tau, it is so on. Function is the same thing. This is the genes family. And another thing, you can say this. You beta, you can say is the genes family. And here you can say alpha 1, alpha 2, and 2, and it is so on. You can say in the same DNA sequence. So at the times this is the alpha cluster. Alpha cluster, you can say same DNA sequence. Okay. So at the beginning of the study the molecular biology, we meet many kinds of the definition. Here, my students today focus on the definition. Understand this. Today we stop here. Okay? Class is over.
Okay,
Okay,
老师
老师
，Thank you。
Thank you, 老师。Thank you, 老师。